all the so-called classic five. So that's the auxins, gibberellins, cytokinins, abscisic acid, and the last one we were looking at was ethylene. And so I think we saw this. This is where we ended up. And so this is the gaseous hormone of the classic five. This is the gaseous hormone. All the rest are not gaseous. They've since discovered other hormones that are gaseous, but we don't have time to get into all the different hormones. But here we're looking at the idea that once you wound a plant, like a stem cutting, for example, that you're going to do on Thursday, the damaged area will start to release ethylene. And it can kind of do a domino impact where it'll start to ramp up its ethylene production. That's what it means when it can trigger its own. Once it gets produced, it'll just keep kind of ramping up. You can use it to ripen up fruits. I've done that a couple of times. If you've ever gone to the grocery store and get a green bunch of bananas, and that's your only option is green bananas, you can go home, cut up an apple, and put that in with the green bunch of bananas into a bag of some sort to keep that ethylene around it, and then it'll ripen up. So bananas do respond to ethylene around it like that. And so many, many times I've used that trick to hasten up the ripening. And then the cut flower industry, if you own a flower shop, one of my good friends owned a flower shop for many, many years. And so she knew of this trick. When you have cut flowers in your refrigerator in your shop, it's releasing ethylene because those are damaged flowers. Really, the stem has been damaged due to the cutting. And so they usually have, they have the cut flowers in a bucket of what looks like water, but really it's typically a solution that is, in this case, it's labeled STS. You don't need to know the name of the chemical, but basically it stops ethylene from functioning to over ripen the flower in this case. So as you can see, the one on the right did not get that chemical, and you can see that the flowers died much more quickly. The other ones look much better. So your profit line is on the at risk here, because if you sell flowers to somebody and they die very quickly once they get them, they're probably not going to visit your shop again. And so you want to make sure that ethylene is kept at a very low concentration in your shop so that you don't have premature aging of the flowers. And so most people, flowers are essentially modified leaves. And so this idea of leaf senescence, you want to try to minimize that if you're giving somebody flowers for any occasion, birthday or Valentine's Day or whatever. And then the second bullet point, auxins and cytokinins. So the two other hormones can stimulate ethylene. And so we tend to see that. So with the fall season, which we've passed through now, but last fall, when you saw the beautiful leaves on the tree, that's partly what was going on is you had auxins and cytokinin concentrations in the leaves starting to fluctuate, and that triggered ethylene to kick in, which then caused leaf abscission and the leaf fell off the trees. So we got to enjoy the beautiful colors first, but there was a lot going on there. And part of the other thing that's going on that we don't see, of course, is the all the nutrients in the leaf tissue, when they're still green, they're siphoning that down mostly to store into the roots before all the color change that you start to see in the fall. So they're trying to salvage everything and everything they can of any use before they start to go through leaf senescence. Okay. So that was just like a taste of what hormones can do for the plant. It's very complex. There's a lot more to it. There's more hormones that they've discovered since the classic five. But it's at least an introduction to get you thinking about this idea of hormones. Now we need to think about how some of those hormones are acting to help the plant respond to its environment a little bit more. This is a book written by Charles Darwin and his son Francis. They were fascinated by plants that they could see moving in their timeline. Plants do move, but sometimes they move so slowly to our eye, it doesn't look like they're doing anything. And it often looks like they only get moved by the presence of wind or, something knocking into it, but they are moving. It's just with you, when you think about some of the plants that they like to study, um, they were fascinated by their very rapid movement that we can sometimes see in some very interesting plants. But this brings up the question, how do they move? They don't have muscles. So how are they doing this? And especially when you think about the quick moving plants, the ones like the Venus flytrap or something like that, you can see it, you can see the trap close in a matter of seconds. So they don't have muscles. They must be doing something to allow for that quick movement. It's something that we've already kind of talked about when we look at stomata, when we said that, the guard cells guard that pore, and the guard cells can move away from each other or they can fold up and seal the pore. And so that's part of what we're thinking here. Basically, one good way that they move is through turgor movements. Turgor is referring to water pressure. And so those guard cells, uh, really what they do with a plant is messing with how much water is in the guard cells, and that will either cause the pores to open or close. Now, the way guard cells are designed, they so if my hands are the guard cells and the pore, so right now they're closing over the pore. The pore is 100% closed. In this situation, the guard cells don't have much water in them. They've gone flaccid, which means they've lost turgor pressure, and they will kind of seal up to each other. Then if the plant says, yeah, I want my stomata open, I want that pore revealed, what they do is they create a situation that allows water to rush in to the guard cells. And when that happens, they get really rigid and they start to move away from each other, and it reveals the pore. So you have to actually get a lot of water into the guard cells to make the pore visible. It's kind of weird. When I first learned this, I had predicted just the opposite. But water rushes in, the pore gets revealed. Water rushes out, the guard cells go flaccid and seal over the pore. And so turgor movements, it can be very, very quick if they choose to do so with the guard cell movement. And so when you hear the word turgor, we're thinking about water pressure. Often we see an asymmetrical issue that's being shown on the right here with that picture. So you have, this is the base of a leaf. And I don't know if you can see, well, I guess you can see, now that I have my glasses on, you can kind of see how the section on the right here that I just circled in red, that is flaccid. There's not much water pressure there. And then the stuff I'm circling now on the right side has filled up with water. All those cells have filled up with water. And so you get this, in this case, it allows for this part of the plant to bend a little bit. In this case, it's bending, it looks to the left. And so that's what I mean by asymmetrical. One side is less water, one side has more water. And so you get that asymmetry, but it causes this bending ability. And so the fancy name for this tissue that we're looking at in the right picture is called the pulvinous region, pulvinous tissue. So I would like you to know the pulvinous. That is basically it's just a special area of parenchyma cells that are in the position of the plant body to allow for this asymmetrical turgor pressure, and it allows for that part of the body to move quite quickly. I'll show you a little bit more about that in a minute here. So turgor movements. That is one way that they can move, often very quickly and dramatically. Another way that they can move, which is not quite so dramatic, is growth. Just the act of them growing and laying down new cells and tissues is a way for them to move. Probably the easiest way that I think about this is roots. When roots are growing and foraging through the soil, it's root growth that's allowing for that to happen as they move through the soil. As the shoots are growing and producing new leaves and branches, that is movement. It's just a growth movement. Again, you can often see an asymmetry to this growth. So we already talked about that actually when we were looking at how Charles Darwin was studying how plants bend toward unidirectional light. That's actually a growth movement. And so we'll look at that again as well. So the idea of gravitropism and phototropism is this idea of asymmetrical or different rates of growth in the stem or in the roots, typically. One of the early experiments that Charles Darwin did is this one here, where no fancy equipment. And what they did was they put carbon, like soot, onto this paper and held it planted above the spiny plant. And then they watched the plant grow, and then the tip of the plant would touch that carbon paper and actually make a tracing pattern as it's moving. And so this is actually, they name this nuttation. They call it a nuttation movement, where you get this spiraling movement. And so if you are good with videography and you videotape a plant growing and then speed it up, you can see many plants, especially vining plants, will do this circling pattern, and they're searching for a support of some sort, because if they're a vining plant, they need to latch on to something and wrap around it so that they can keep upright and get access to sunlight. So again, pretty simple, no fancy technology here, but they were able to demonstrate that plants do this very definite movement. Here you see nodding movements, and well, you don't see it happening because it's a picture, but basically, if you think about a seed in the soil and it's germinating and growing, what's the first thing that comes out of the seed, by the way?
Radical.
Radical. All right. So the radical comes out and starts searching for water, and then the shoot comes out and starts to want to get above the soil, right? That's the goal. The shoot needs to get out. And so if you are a plant, baby plant trying to do this, if you just try to go straight up through the soil, it's not as efficient. What they've actually noticed is that the baby plant is busy nodding back and forth, swaying back and forth as it grows and moves up through the soil. Because you can imagine if you're buried in soil, punching straight up would be a little tricky, but if you kind of move around, it helps the soil particles move away from you, and it's a little easier to make your way up through the soil. And so they actually do see that. In this case, what they did was instead of soil particles, they have those little gel beads that they planted the seeds into. And so when you're studying this, I have a video of this. So if you plug that in and use it for study tools, you'll see that nodding movement. They'll show it to you. And so it makes sense when you think about it. It's like, oh yeah, that seedling does have to get through the soil. It helps to be able to break the soil particles apart and away from each other. Tropism, gravitropism, phototropism, when you see that word, it's I don't know, permanent. I would put that in parentheses. It's movement resulting from external stimuli. Um, you can, yeah, I guess permanent. I see what they mean by that. But anyways, it's movement resulting from external stimuli. And so it's, either the plant is attracted to something and is going to grow toward it, like light. If you're the shoot and leaves, if you're the roots, you probably aren't attracted to the light and want to get away from it. And so they call it, positive phototropism if the leaves and shoots are growing toward it, negative gravitropism if the roots are trying to hide away. For a root to be exposed to sun, light, it's not normal, at least for most plants. Other plants, there are plants that hang out on branches of trees where their roots are exposed, but we're not thinking about those right now. Okay, so the average plant doesn't want to be in sunlight because that's telling it, I'm above ground. That's not where I'm designed to be. I'm designed to be below ground where I can take up water and minerals. In fact, they're now starting to rethink this idea. A lot of research, when you do research on plants, you want to look at root growth. And so they're exposed to lab lights and sunlight. And now they're saying, you know what, we've been doing that for decades, and that might have been a variable that's been messing with our experiments because that's not what roots are designed to do. And here we are putting them in this artificial situation, and they may not be behaving true to themselves, so to speak, when you are exposing them to light. And so now they're rethinking some of those ideas, which I think is a really good observation, and it's a smart idea to think about that carefully. So here you can see the plant is kind of tilted, all of its leaves, even the flower is tilting. It looks like must be the light is coming from the left, is what I picture. So that would be positive phototropism, or often we just say phototropism because we tend to think about the shoots and leaves for this. And again, it's that auxin hormone we talked about last week, where we were saying how it's a growth stimulation, asymmetrical growth stimulation of the stem tissue, right? We say that unidirectional light hits the shoot tip, it shifts the auxin in the shoot tip over to the dark side, and it starts to course down the dark side of the stem. So that's auxin coming down in the blue. And it's telling all those cells grow, 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 grow. And so they're going through cell division, they're elongating a little bit, and the lit side, so the light's coming from the right in this case, all those cells are like, no, we don't need to grow so much. And that's how you get that bending pattern. So that's a growth movement. You have asymmetrical growth on that stem that causes that bending pattern. Does that make sense? Okay. So here you're looking at root tissue and gravitropism, this idea of the roots want to grow in the direction of gravity because that's telling them that's their best option to get down into the soil, is to grow down toward gravity. Oh boy, there's been a lot of debate about how gravity is perceived by plants. Interesting debates in the literature on this, but basically what they're saying currently is that it's kind of a situation here where, so this is the root tip area, and all these little spherical blobs in these cells, those are amyloplasts. So you looked at that in lab, right? You looked at amyloplasts with potato tissue, probably. What do amyloplasts store? What kind? Starch. Okay. So these are roots are usually chock full of amyloplasts because they're just a really good place to put nutrients for storage purposes. And so here you have a lot of amyloplasts. And do you see how they're not scattered evenly through the cell? They're like settling and have sedimented to the so-called bottom of these cells, right? So what they think is going on, I'm trying to show it here too a little bit more dramatically, how the amyloplasts are settling due to the force of gravity. So this root's growing straight down. The amyloplasts have settled, and what they're doing is all those amyloplast organelles are creating pressure on just that part of the cell membrane. And so this part of the cell membrane is getting more pressure from those amyloplasts compared to any other part of the cell membrane. And that's how the root is determining, oh, gravity's this way. I'm feeling pressure on my cell membranes equally on, the very bottom of my membrane of each cell. And that gives them the signal to go ahead and keep growing in that direction. Yes.
How does it know where the gravity is and which way it's below them? Like, could it be facing it towards, and that is like that processes it by?
Yeah. So if you put a root on its side, do we have a picture of that? Yeah. Let me show you this picture. So if you put a root on its side, these are supposed to be the amyloplasts. You see how they come over this way instead of being right here like the previous image, because the root was going down, it's all amyloplasts here. Now, when you lay the root on its side, they shifted. And so now there's pressure here, more pressure pressing against the cell membrane portion than anywhere else. Yeah.
So hypothetically, if you were to grow a plant upside down with a light source underneath the plant, what would win with gravitropism and phototropism?
Depending if the water is above it, would it go up? If there's water above it?
So if we turn the plant upside down and was watering, so the soil, like a potted plant, and we turned it upside down, you're asking, would the roots start to grow this way through the?
Light source underneath. Would the plant grow down and the roots grow up then, or would it still try to reverse itself?
Has anyone grown those? Don't they have pots where you grow potatoes that way? There's something I thought I saw, a gardening bag or pot or something where you grow plants upside
I've
down.
grown tomatoes upside down.
Maybe it's tomatoes I'm thinking of. Did you notice?
They just kind of stick to kind of the bottom, so the tomato will still like grow down, but like that picture will still grow out and up.
The tomato
The
stem.
actual stem will grow
Mhm.
out and up. The
Mhm.
roots just kind of stay towards the bottom of it, but they will sometimes go up
Mhm.
towards the water.
Yeah. So the stem, if the stems are dangling initially, over time, if they can handle it, they probably will try. Well, that's with normal sunlight. If you put a light underneath it, especially a nice bright light, they probably would grow straight down for that. If you have like a sunlight garden, then they will try to track the sun. And then in lab, the propagation you're going to do on Thursday, one of the plants is called a snake plant, snake vine plant. And that's just a leaf, bunch of leaves coming out. And if hopefully we have one on the table for you to propagate if you want. So you'll take a leaf cutting, and if you accidentally turn it upside down and plant it that way, you'll get roots coming out of the top instead of, so it's like you fooled it. And it's like it doesn't know. It's because you planted it upside down. If I can find the right PowerPoint at the end here, I'll try to pull it up to show you. So sometimes you can fool plants that way. But there's been a lot of debate about this. Like, so whoops, this idea of the course, let me go back one. They had to give it a fancy name. They call them statin. The statoliths are pressing against the membrane. They're just amyloplasts. That's all they are. I'd rather you know that they're a bunch of amyloplasts. It's not like there's some magical thing. They're just a bunch of amyloplasts, but they had to give you another word to memorize. So, and you can see here, they put this potted plant on its side, and you can see over time, but this is where the light is coming from above. And over time, the plant is moving its shoots and leaves up. So partly it's hormone development too in this case, because, and I'm not going to go into detail, there's a lot to this, but basically what you see happening is if you have a root growing straight down, like this top image, you can see, so IAA is a type of auxin hormone. So the auxin is coming, because the root is straight up and down, the auxin is coming down, straight down. And then what it does is when it gets to the root tip, and this is kind of a new discovery, the auxin comes down, gets to the root tip and does this, they call it the water fountain effect. It'll kind of course back up the outside. That's what they're trying to show with these arrows, that it comes down and then it starts to course back up the outside of the root in equal measure. Okay, so it's everything's very equal. And then they show those amyloplasts pressing down, and so that root knows gravity this way, I'm gonna grow this way. And then if you take a root and lay it on its side, now you see auxins coming down, and instead of equally going back up, you can see by the arrow, they're trying to say that there was more auxin on this part of the root compared to this part of the root. And it's weird because if you have a lot of auxin in root tissue, instead of speeding up growth, it tells the root slow down growth. So this is a really good example of how gnarly it can get with hormone research. I just got done telling you when phototropism happens, the stem, in the presence of auxin, the stem tissue is grow. Now I'm telling you it's saying don't grow. And so it's because you have two very different tissue types. Instead of a stem tissue, you have root tissue. And so hormones, they've learned, can act a little differently depending on if it's in the stem and leaves versus the roots. So keep that in mind. It's not necessarily going to act the same way in every single part, especially when you think about above ground, below ground tissue. So this is weird. It actually tells this side of the roots, slow down your growth with that much oxygen. A little bit of oxygen actually tells it, go ahead and speed up. And that's how you get the growth movement to allow for it to bend toward the gravity force. So it's complicated, and there's been a lot of debate in the literature, lots of different experiments trying to tease this apart. But they do feel that the amyloplasts are the pressure exerted by the amyloplasts in different parts of the membrane does help tell the plant where gravity is, and then the auxins will stimulate the roots appropriately to grow toward the force of gravity. Okay, here's another one. Thigmotropism. That word is, thigmo means touch. Thigmo, touch. So this is touch response, growth in response to touch. So the classic example would be a tendril. Like if you think about pea plants, if you've ever grown peas in your garden or beans, and they have those these vining tendrils. And if you've ever grown lots of, sometimes when we have a lot of bean plants growing on our grow light in the lab, you'll start to see them twining around each other, and it's like, oh man, and you try to separate them without damaging anything, and it's a pain. So be careful with your beans and peas, because they can cause problems. But basically what they're doing is they're sending that tendril up and they're moving. You can't necessarily see it in your timeline, but it is moving. And once it latches onto a support, now its tendril is touching the support. Maybe it's a garden stake, and it knows it's touching that garden stake. So thigmotropism, and then that triggers, in this case, the vining portion of the plant to start to wrap around the support. And so that's a classic example of what you're seeing here is a growth response. What actually is happening is the part that's touching the stake will actually slow down its growth, and the part that's not touching the stake will speed up, and that causes the coil. And then it'll make another coil around the stem, and any part that's of the tissue that's touching the stake will slow down, the other part speeds up, and you just keep getting that growth response that causes that coiling around, wrapping around. That video, when you're studying, will show you that. So morning glory plants, pea plants, bean plants, that is very common. And there's a whole lot going on there when they're touching that branch that they're coiling around, or the garden stake. There's a whole lot of gene expression and shifting of asymmetrical growth going on, so that, you know, that the part that's not touching, like this part up here, has sped up its growth, and that causes it to coil around like that. So it's pretty effective. Seismonasty. What does a seismograph measure? You don't really have this issue around here, but California does. What is a seismograph? Earthquakes. Yeah. So if you can think about that, so nastic is movement. It's another way to say movement. So it's a response to mechanical disturbance. And so these pictures you may recognize. What's this one? Venus flytrap with a 4 hapless fly. What's this one? This one's a really petite plant. You have to really search for it in a bog. They're hard to find. Sundew, all right. If you're lucky to find them around here, you have to have a really good eye. How about this one? Sensitive plant. Yeah, the genus is Mimosa. That's actually quite easy to grow, by the way. Just be kind to it. Don't touch it all the time. You're making it tired. All right. But anyways, so these are the more dramatic movements that Charles Darwin was very fascinated with. And so what you have going on here, so Mimosa pudica is the name of the sensitive plant. What you have going on here is the turgor movements that cause very rapid shifting. Here it's a modified leaves that we call the trap. It allows it to trap the insect And same with here, you have the sundew leaf being able to coil around and capture the insect crawling around. Um, So very, very fun. So this picture is showing Mimosa pudica, the sensitive plant in its native habitat. It likes to grow along roadways in tropical areas. And it's mixed in with other plants here. So let me show you this picture, and now I'm going to show you the next picture. Do you see the difference? Let me do it again. So your mimosa are these delicate looking plants with all these little leaflets. Okay, there's one with the pom-pom flower. Now watch again. Do you see how they've all closed up? So when they did it before or after, where somebody must have come along and brushed across that section to take the picture of the after. And so I mean, it's a nice name, sensitive plant. That's very good name for it because just touching one of those leaflets can cause the whole leaf to start to close up. And there's video for you to look at, a couple of options for you to do when you're studying, but very dramatic, very quick. Why would you do this if you're mimosa? I mean, don't really think about a pencil, but why would you choose to do this?
So you don't get eaten.
Yeah, so if that pencil was a fly or a grasshopper or a moth or something, you wouldn't. I still can't find it, but I did see video footage a while ago. I think it was Sir David Attenborough video where they showed a beetle crawling around on the mimosa. And of course, if you've ever watched, we will watch Sir David Attenborough in this class, by the way. But put to really cool music, and then you see this beetle walking along, and the plant does this, and then all the leaflets on the whole leaf close, and then the whole leaf bends, and the beetle just tumbles off, and you swear the beetle's like, "What just happened?" It was so dramatic, but it was a very nice video to show that's a good way to confuse and get your predators off of you.
Are there hormones telling the plant to change the water pressure, or is that just something that happens?
They don't know for sure. What they do know for sure is that it's rapid turgor movement. Now, they are searching for the hormone issue. I wouldn't be surprised if there is some kind of hormone involved, but right now they're just showing that the plant plays with how many solutes is in the pulvinous region, and that causes either uptake of water or expulsion of water in the tissue. And that's what's causing the very rapid movements.
Branch of or is thymotrophism like extended periods of time that the plant is touching?
Yeah, so thigmotropism is the slower response of, like, think about a pea tendril, wrapping around a garden stake. This, let me go back to the seismonasty term is more from an outside disturbance, like a fly landing on that, or my dad's dad was 100% Finnish, and we'd go to visit my grandfather. He lived in Wakefield, Michigan, and I remember climbing a tree and watching my grandpa's neighbor go into his backyard with a baseball bat, and he was whacking at the tree trunk, speaking Finn. It was that whole neighborhood, there was all Finn, so they only spoke Finn. So I couldn't tell what he was saying, but he seemed a little angry. And I remember talking to my dad about that afterwards, and my dad was saying, "Oh, the neighbor," what my dad told me was, I was really young. He says, "Oh, the neighbor was doing that to make the tree tough." I later found out he was really mad. He got in a fight with his wife, and he was like venting, which was at least a safe way to vent. And actually, now that I learn more about plant phys, he was doing this. He was hitting the tree trunk, and if you were to do that enough, you would actually change the overall shape of the plant. Well, it's not so much seismonasty, probably more, there's another term I'll introduce here in a minute. But yeah, it's this, what I'm trying to get across is it's an outside like mechanical disturbance of some sort. You can do it too with sensitive plants. If you put them on like a shaker platform and shake it, you can see that it will do that movement too. So it is a little different than the thigmotropism. Plus, this is turgor movement, whereas the thigmotropism is growth movement. So that's a big difference too. Okay. And then again with the Venus flytrap, they show some cool video of that. So this is, they got little trichomes in here in the modified leaves that are the trap. There's little trichome hairs in there that when the insect is crawling, if they trigger two of those hairs, that's the signal for the trap to close over. That's why they say plants can count. They know they can tell the difference of if only one hair has been triggered or if two. And if two has been triggered in under 20 seconds, they'll close. If it's because you can imagine if you're a Venus flytrap plant and a little twig lands in there because of a wind blowing a piece of twig in there, you don't want to digest that. That's not something you want. And so you won't close, you won't bother going through the, energy to close over that. You'll just stay open. But if an insect's crawling along, you want that. By the way, why do Venus flytrap want insects? They don't really care about twigs. They want insects. What is it about the exoskeleton of insects? It's very high in a particular element that boggy areas seem to be low on. If you've learned anything about bogs. So bogs are really low in nitrogen. Exoskeletons of insects really high in nitrogen. So it's a awesome adaptation to living in an area where they can't get enough nitrogen on their own from the boggy environment. So they get it from the insect's exoskeleton. So I've had this question many times over the years. If I buy a Venus flytrap for my house, do I have to go catch flies and feed it? Which isn't the most appealing thing to do. No, you don't, because the growing media they put the Venus flytrap into is nutritious enough. So you don't have to. You can if you want. It's fun to watch. When I was trying to brainwash my boys that plants are cool, I bought a bunch of these and we lived in a house that for some reason, have you noticed this? Depending on where you live, sometimes flies just congregate in like the windowsills. So then I grab one that was still alive and drop it in there and get them all, fascinated by that. And then my cat at the time was a really good fly catcher. He loved to hunt flies. And so we get him going on it and he would grab the fly and stun it and drop it, but he wouldn't eat it. So then we'd grab it and put it in here. And yeah, so it was a team effort and it was cool. They thought it was cool. The other thing my boys would do when they were toddlers and they got really, really mad at me, they wouldn't like yell at me. They'd like take a flower off one of my plants and rip it apart. They knew that would get me. Little stinkers. So yeah, they would kind of worked against me in that case. But anyways, beautiful sundew. So again, the videos are really cool to watch as you're studying up close to the sundew. They grow really low to the boggy. They're usually kind of embedded in sphagnum peat moss, so they're really hard to find in bog environments. So if you find them, count yourself lucky. Okay, we'll stop there. So we'll get into some other movements and then we'll look a little bit at how plants respond to light. We have to think about that a little bit more.